and he's going to be talking about differential gene expression and some marsh plants.
Thank you. How's it going, everybody? Thanks for coming. My name is Mariano Alvarez. I'm a fourth-year PhD student at the University of South Florida in Christina Richards' labs. I'm looking for a postdoc right now, so. And today I'm going to talk about the transcriptional response to the BP oil spill in Spartina, alterniflora. So in April of 2010, the Deepwater Horizon oil spill released 4.9 million barrels of oil into the Gulf of Mexico. For our European friends here, that's 780,000 meters cubed. And that oil made landfall all across the U.S. Gulf Coast in Louisiana, in Mississippi, and in Alabama. And it impacted the salt marsh communities along the coast there, which are primarily comprised of Spartina alterniflora, it's the foundation species of these marshes. It makes up to 90% of the biomass in these marshes. And it really affected the Spartina. You can see here the dieback right here where the oil washed up and killed all the plants here. But this picture is taken about 3 months after the oil spill first started. So you can see already, it's a little hard to see on the screen, there's some regrowth there along the edge and up and through. And that's not really all that surprising because Spartina alterniflora is used to a really stressful habitat. So the salt marsh has this really long ecological history of being this really heterogeneous really dynamic environment. And Spartina alterniflora lives across almost the entire salt marsh. So it has a lot of stress that it deals with on a daily basis and a lot of mechanisms to cope with those. So it's dealing with two types of stress, this chronic stress that happens all the time, which are things like tidal cycles, the nutrient flows that go in and out of the marsh, and this really strong salinity gradient from the high marsh to the low marsh. And it also deals with acute stressors like oceanic storms. In the North Atlantic, it can experience ice flows, which scrape the marsh bare. And also chemical spills. So the Gulf Coast is pretty active for the oil and gas industry, so it's not the first time they've experienced oil. So I was really interested in how the Spartina was dealing with this oil stress, how it was able to rebound and recolonize those areas. So I wanted to look at the mechanisms of the stress response, and usually we think about evolutionary mechanisms and physiological mechanisms. So what I mean by that, when I talk about evolutionary mechanisms, I'm talking about things like selection and adaptation and divergence. And when I'm talking about physiological mechanisms, I'm talking about things like reduction in above ground biomass, which is one of the mechanisms that Spartina uses to deal with stressors. Also changes in water use efficiency. But because we have molecular biology techniques now, we can look at molecular mechanisms too, and really ask what is the molecular basis of this stress response? How is this response constructed on this very fundamental scale? So molecular mechanisms are things like protein regulation and gene expression regulation. And I'm going to talk about the second, gene expression regulation today. So I just looked at transcription, but I briefly want to talk about why I'm looking at transcription. So the first and most important reason, particularly for this venue, is that variation in transcription is heritable. So it has evolutionary consequences. It's also sensitive to environmental stimuli on both shorter and longer time scales. And variation in gene expression very importantly affects phenotype, and it does that through changes in protein expression. And this is a little animation of transcription right here. Sorry if it's hurting your eyes. So we chose to look at the whole genome expression profile using a microarray. This microarray was designed with our brilliant collaborators in Rennes, the Anouche lab in Rennes, France. We pooled our RNA samples when we hybridized this microarray. And the transcripts on the microarray were annotated from literature, from Oryzis sativa, which is rice, it's a fairly close relative, and Arabidopsis thaliana, which is our model plant. I do have a slide here on the design of the microarray, but I won't go too much into detail, but if you do have questions about it, feel free to ask me afterwards. So the design of our experiment was as follows. We had 3 locations, so I realize there's only 2 dots on this map. That's because this site right here is actually 2 sites. They're just geographically fairly close. They're both in Barataria Bay in Louisiana. And our 3rd site was up here in Bay St. Louis in Mississippi. At each of these locations, we found an oil-contaminated site and a nearby uncontaminated site, and we assessed that through the visual presence of oil. So you can sort of think of the design as like a split plot design where we have treatment and control replicated 3 times. And we took 9 samples from each site, I should point out. So the analysis was pretty straightforward. We normalized the data, we did principal components analysis, and then we did a pretty standard analysis of variance. So I'll show you the principal components analysis right now. You can see here on the left, this is PC1 and PC2, and we get this really nice separation of our oil and no oil sites. It looks really good. We're really happy, except for Mississippi. The Mississippi site is grouping with the oil sites. We're really not sure why that is. It could be that there was previous contamination, historical contamination, or it could have been that there was oil contamination there that we just missed somehow. But either way, when we introduce PC3, we get this nice separation of the oil and the no oil sites, and the Mississippi sites up top, and the 2 Louisiana sites down at the bottom. So then we did an ANOVA, and we included state in the model just because we were concerned about the geographic distance between the sites, but we didn't have too many genes responding to state. So what I'd really like to focus on is this 3,622 genes that are responding exclusively to the oil stress. So we were pretty excited about this result. Now we have these genes, but now what? How do we make sense of these 3,622 genes? Which ones are more responsible for the response, and which ones are just sort of downstream? So to look at and try and decipher which ones are more important, we decided to put them all into an interaction network to look at which ones may be regulating the response. But when we put them all into an interaction network, we came up with something that looks like this. It kind of looks like a hairball. It's really not that informative. So we had to find a better way to digest this and break it out into components that we could really, that had biological meaning. So we used a gene ontology analysis. I won't talk too much about it except to say that I'm sure most people are familiar with it, and it describes gene function in a hierarchical way. And we use annotations from Arabidopsis thaliana, so we only use genes that had homologs in Arabidopsis for this part of it. So we came out with this, with these series of gene ontology categories, and this big one over here, this green one, that's cellular process, that's not too detailed. Metabolic process, that's also not too detailed. But we were really interested in this blue category over here, which is response to stimulus. And we thought that that might be the category where it was most specific to the oil response and not really just the downstream stressed out response. So we have subcategories in here that we looked at, one of which was particularly interesting was response to cadmium ion. So when the oil comes up out of the ground, it's pulling with it a lot of heavy metals, including cadmium, so we thought that this might be a really good indicator of how the plant was responding. So we modeled we put all the genes in this category into a biological network, and this is what we came up with. It's a little bit more interpretable. These genes in green, each of these dots is a gene, and the ones in green are genes, all the genes in Arabidopsis that are annotated as being part of the cadmium ion response. And the ones in red are the ones that responded experimentally in our study. And we can look to see which ones are more connected than other ones, which ones are sitting deeper inside this network. And our thought was the ones that are more connected are more likely to be orchestrating the response, whereas the ones sort of out on the end might be less instrumental in the response. So we're really happy with this. Now we have these genes that we think are interesting. But how do we know that these genes are actually really responding to the oil? So these were natural populations living out in the wild. And we don't know if the response that we were seeing was really oil or whether it was some artifact of just being in a really heterogeneous environment. So we decided to bring everything back into a controlled environment. We did a follow-up study where we exposed oil-naive plants to oil from the BP oil spill. And it was pretty straightforward. We just had a treatment that got 2.5% oil and experienced 1 tide per day, and a control group that didn't have any oil treatment. And we're using qPCR right now to look at the expression of our genes of interest, both to confirm the microarray and also to confirm that the response is really due to the oil. And I'm actually running those right now. I just ran one before I left. And looks like we do have the same differential expression that we saw, but I don't have the results yet, so I won't tease you with it. So where do we go from here? So we have these genes that we think are really interesting, but we want to know that these genes are really causing the phenotype that we're seeing, which is a recovery after the oil spill. So to say that these genes are really causing this response, we wanted to have a better type of validation that isn't just correlative. So we're using knockout lines in a closely related species called Brachypodium tachyon. It's another model species in the Poaceae family. And we're knocking out genes that we identified from the microarray, and those are growing right now. We also wanted better phenotypes other than, survived the oil or didn't survive the oil or, wasn't exposed to oil. So we've recently gotten access to a light core, which is really magic. We get this really great data about photosynthetic rate, water use efficiency, and we also wanted to understand how this differential transcription is occurring. Is it due to SNP variation, or is there actually a selective event happening in the oil spill? Or is it something like, is it some kind of phenotypic plasticity and possibly mediated by DNA methylation or some type of other epigenetic marker? So really quick, I just want to toss out, we have a technique that we're using called epigenotyping by sequencing. I highly recommend you check out Thomas Van Herk's talk tomorrow morning. He will blow your mind. But this is just a taste of the data we're getting from it. And it looks like I'm out of time, and I don't know if I have time for questions, but thank you so much for being here, and I'd be happy to talk with you afterwards.
We're good on time. We have like 5 minutes for questions if anybody has any.
Well, great. Maybe I'll also thank the people who helped me then. So I don't look like a jerk. Thank you to my lab mates, Marta Robertson, Christy Faust, Holly Kilovitis, and our honorary lab mate, Enrique Saez Laguna, for their awesome feedback. Thank you to Brittany Deloach McCall and Steve Pennings for helping us locate the oil-impacted sites and not letting me get eaten by an alligator. Thanks to Koen Verhoeven and Thomas Van Herk for the extensive FEGBS help and the general brilliance. And thank you to NSF for funding this grant. So I'll take questions now. Yeah.
Do you found any gene or group of genes that may be insulated with DNA marks of mutation in response to oil?
I'm sorry, could you repeat that?
So when you got like your different hematology groups, my question is that did you check if within those groups is there any gene or group of genes that may be involved in establishing different epigenetic marks in the genome?
Yeah, so we did see the differential expression of few methyltransferases, so that was pretty exciting from the epigenetics front. So we were hoping to see some of those genes in our epiGBS to see if there were SNPs in those sites, maybe selecting for variation within those methyltransferases. It looks like we might also have differential methylation of the methyltransferases, which is a little bit meta for me. But yeah, we definitely saw some response in the methylation machinery, so that was really encouraging. Yeah.
Could you explain in a little more detail how you interpret that transcriptome network figure that you?
Sure, absolutely. So let me just go back a little bit. So I'll go to this one, which I think makes the most sense. So for this one, we were just looking at number of connections. So we just chose genes of interest purely based on how many connections they had within this network. I will say that I went to some really awesome talks, I think yesterday, and I learned about something called eigenvectors, which is connections of connections, so genes that are connected to other highly connected genes. So I'm hoping to get a little bit more sophisticated with this network analysis. This is sort of our first stab at it. But if you have any ideas about how to analyze these networks, I'm totally open.
What do the linkages in this network represent?
So these linkages are based on actually a few different types of annotations. They're based on KEGG annotations and Aristide annotations, which are metabolic interactions. They're also based on the regulation of transcription and protein-protein interactions. So they're really all in there. So really, we just took any type of interaction from this network, but we could pare it down by the type of interaction